Hi guys, my name is Joe, and I have tasked myself with creating a series of videos that allow you to decide whether prions, which are proteins found in the human brain, are friends, or conversely, are they foes? Like a what?
An ion.
No,
Is
a
it
prion.
an electron?
A prion.
A pretty.
It's the one before an on.
A prion. A prion.
Stop filming me.
Do you know what prions are?
Prions? Never heard of them.
Hyman. What are prions?
What?
What are prions?
Prions?
What are prions?
What is a prion?
Any idea what prions are?
Prions. Let me tell you. A prion is a transmissible amyloid. Don't worry, that's not supposed to mean anything at this moment. You're not supposed to think, thanks Joe, I now know exactly what a prion is. But bear with me and let me explain. Now, of course we know about proteins. We know, or maybe don't, that they are simply a chain of amino acids in a certain sequence that ultimately decides its overall structure and conformation. So, we know that proteins fold into lots and lots of different conformations, way too many to count, and this makes them versatile. They can be enzymes, receptors, anchors, and even transporters on phospholipid bilayers of cell membranes. When a protein folds into a certain conformation, however, it is able to cause disease. An amyloid, as shown here, is a protein deposited in certain organs in certain diseases which stop it from working properly. Generally, proteins in their functional state are soluble, such as an enzyme, and can be easily broken down. It is the beta-pleated sheets that form which make the proteins insoluble and impossible for the body to get rid of. Well, I'm sure you'd agree so far it's some fascinating and mesmerising stuff, but where does the name of the video, how do prions all relate to this? Well, as we alluded to earlier, they are transmissible amyloids. They are able to do everything an amyloid is able to do, however they are transmissible, which means it can pass between organisms. We'll discuss exactly how prions are able to be transmissible later on in the series. It's very important to remember that prions are made from protein, and they have a functional state. They start off life as healthy and functional, and are found in the brain. It is when they are converted they become problematic, and just like amyloids are able to form insoluble aggregates that stop the brain from working how it should. It is the beta-pleated sheets we talked about before that cause the insolubility. It is the ability for prions to cause aggregates and prevent the brain from working properly that causes the argument that prions are a foe. Yet, I won't ruin the next video for you by elaborating on this. Instead, let's talk about why prions have had scientists scratching their heads even more after every development in their structure and behaviour. Watson and Crick won the 1962 Nobel Prize together with the ever-forgotten Maurice Wilkins in medicine for the discovery of the structure of DNA. This structure was the missing piece in the puzzle in terms of DNA replication. It led to the popular belief that DNA is the central reason for the passing on of traits in a Mendelian fashion, and so is the central reason for any replication within biology. Work done by three scientists, Tigver Alper, I.H. Patterson and J.S. Griffith, all discredit this belief. Alper is the one shown on the screen. Pictures of the other two are hard to come by, but I'm sure you'll agree Alper's infectious smile more than makes up for it. It was known that prions spread and propagate so that proteins fold in an identical fashion. It was thought for this to be possible, DNA must be present. And don't be scared by the picture. It's literally just to show how prions are able to spread throughout the brain in a prion-like fashion, and this will be explained in detail later. The scientists went about carrying out a host of complicated experiments. The basic aim was to attempt to inactivate the activity of prions. This was done by using nucleases, an enzyme that breaks down nucleic acid and essentially chops up the DNA into smaller subunits. Also, the use of UV radiation was utilised. Unfortunately for the group of scientists, the experiments failed to work. Prions were still able to go about their usual behaviour and aggregate. This led to a lot of confusion. How could prions propagate in an identical fashion without the use of nucleic acid and DNA? It wasn't until 1982 that the confusion was put to rest. Stanley Prisner, another smiling scientist, probably this time because of him winning the Nobel Prize for his work, used proteases instead of nucleases. Proteases are enzymes that break down proteins. It was found that with the use of these enzymes, Prisner was successful in inactivating an infectious prion. This led to the protein-only hypothesis, the idea that prions go about its behaviour, replicating itself, spreading through the brain and passing on traits without the use of DNA. The work challenges very popular scientific concepts, such as that there is only one possible explanation of how life has evolved. Are there possibly other mechanisms of heredity in living organisms? Perhaps this is just one indication of how much more we are yet to discover in this scientific world. Now, these are some bold claims, and many scientists believe justifiably that more work needs to be done to clarify the action of prions. Yet, it is undeniable just how revolutionary prions are. Because of the nature of prions, when they go about causing disease, they become particularly problematic. As they contain no nucleic acids, they are resistant to procedures that would usually kill pathogens, unlike bacteria, viruses, and fungi. Also, there is no trigger of an immune response, as a prion causing disease is simply an abnormal version of a previously harmless protein. So, that concludes the first video in the series. I hope by now you can at least pronounce prions, and a real bonus would be that you might actually know what one is. But if you're struggling, and there is a lot of information to get your head around, let me give you a quick summary. If you don't remember anything from the video, try and at least remember this. A prion is a transmissible amyloid. An amyloid is simply a protein folding in a way in which causes disease. Remember, they are insoluble and aggregate when they cause disease. Prions are simply proteins, and they start life as harmless and functional, and then convert to a disease-causing state. We'll talk about why they convert and how they convert later on in the series. A prion is able to replicate itself and pass on traits without any presence of DNA. This is the prion anomaly. Because of this, they challenge basic scientific dogma, and they become problematic when causing disease, and there's still lots and lots and lots to learn. In the next video, we will look at why one might consider prions as foes. But until next time, thanks for watching,